Okay, welcome to Thinking Beyond. I don't have any announcements about our upcoming speakers since this is our last webinar of this semester, but we'll start up again in the fall, so look out for emails from me on social media posts mid-August. And then for tonight's webinar, we will take questions at the end of the hour, but if you have them at any point during the talk, go ahead and submit them using the Q&A feature at the bottom of your screen. And that's all I have, so I will turn it over to the Beyond Center Director, Paul Davies.
Well, hello, everybody, and welcome back to Thinking Beyond. As usual, we've got an intriguing and entertaining subject to discuss today. And we have a guest speaker who I'll introduce in a moment. But I should say that Sara Walker has had to give her apologies. So I will be moderating the events this afternoon or this evening, or indeed this morning, in the case of our guest speaker, who is at the moment, he's coming to us from Turin in Italy, but he's normally in Grenoble in France, where he is professor and scientific director at Fontinatech. I'm probably not pronouncing that very well, University Grenoble Alpes. He's got appointments as well at University College London, that's easier to pronounce, and indeed the University of Turin, or Torino, I guess. He's an expert on the brain and exploring patterns of brain connections and the way they develop. And he's also worked on brain cell survival in neurodegenerative diseases. So brains are important tissues, but the truly astounding work that he's doing now with his colleagues in Grenoble has to do with whether photons, a word that makes all physicists' pulses race, the photons might play a role in not just in tissues around the body, but in the brain itself. And so we're going to hear from John about that. And then we'll be joined a little later on by Nirosha Murugan, and I will introduce her when John has finished his remarks. So, John, over to you.
Thank you, Paul, and thank you for the invitation to speak to you today. It's such a pleasure. And what I'd like to talk about is an oldish idea that seems to be gaining some sort of new life in the last few years. And that's the idea that cells, brain cells, or body cells indeed, may communicate with each other using light. And what I hope to convince you, together with Nirosha a little bit later, is that they may use this light that they generate themselves, not only to communicate, but to repair each other, themselves or others. So what I'd like to do today is firstly talk about how cells communicate, how brain cells may communicate, some of the history behind that. Introduce this concept of biophotons, which is basically the light that the brain cells or body cells may produce. Then talk about a little bit about some of the work that I've been involved in for the best part of 20 years of using external light to help cells function and stave off disease, and how the biophotons idea works with applying this external light. And then move on to some of our early results in Grenoble on the biophotons idea, and then talk about some future applications, and I hope to bring Nirosha in to that at the end. So first up, a little bit of history. It's a classical cartoon of the brain. And of course, we don't really, it's the most serious of organs. And don't be fooled by this somewhat placid appearance. That brain is jam-packed full of cells, all very, very busy bees, and it's one hub of activity. So it's full of brain cells. These cells communicate with each other. It's often regarded, if we understand how they communicate with each other, then we've got a really good idea of how that brain as a mass may work. Now, the history of trying to understand how the brain cells communicate is very and quite lovely. At the turn of the last century, there were two very, very defined school of thoughts. There was the reticulous or the reticular school led by this chap here, Camillo Golgi, and they believed that brain cells formed a continuum. So they were actually connected all together, and that's the way in which they work together. In fact, when I was in England doing my postdoc, my old boss was the grandnephew of Dieter. In fact, he looked a little bit like Dieter's. So that was one school of thought. And the other school was led by Ramon y Cajal, and he is considered the father of neuroscience. And he proposed that really there was the separation, that each neuron was separated and there was some kind of communication through that separation. And indeed, what happened, they got the Nobel Prize together in 1906, Golgi and Ramon y Cajal, and there was an overall at the ceremony, they were almost physically assaulted each other because they were that passionate about their differing viewpoints. As it turned out, Ramon y Cajal was correct. The neurons were separated, and they separate. At that separation, there was a synapse, and that was the point of communication between the cells. And subsequently, since that time, it's been shown that cells may communicate either chemically using substances named neurotransmitters to change each other's activity. Others have shown that there indeed could be an electrical communication, and they're called gap junctions, nowhere near as common as a chemical communication. For the last 20 years or so, various gases have been identified as a means of communication between cells, and the most popular one is nitric oxide. So I grew up when I had lots of hair some years ago. I grew up with the notion that cells can communicate chemically, electrically, or using gases. Now, about 100 years ago, the idea was proposed by this chap here, Alexander Gurwitch. Electromagnetic radiation may be a means of communicating between, and he was pretty much ignored for a long time. And some 60 years later or so, that type of electromagnetic radiation was defined as light, and that was mainly through Fritz Albert Popp, but there were others, but he was the one that really drove this. So that idea has been developed since then, mainly using plants, various types of bacteria and cancer cells, and that's a lot of the work that Naroshi's done over that period. And it's been referred to as ultra-weak photon emissions, like Paul said, or more recently, they've been called biophotons. I tend to like the biophotons term. So what do we know about these biophotons? First up, they're very weak. They're very weak. You don't see them with the naked eye unless you've got special sorts of qualities, but generally most normal people do not see biophotons, and you need a super duper sensitive piece of machinery to detect them. We're only talking 2 to 200 photons per second. That's incredibly weak. And they're associated with very key organelles within the cell. So here we have a typical neuron, cartoon of a neuron, and the main organelle within that neuron is called the mitochondria, and they are the engine rooms of the cell. They produce the ATP energy, which drives a lot of cell function, makes the cell function normally. Also produces an important molecule called reactive oxygen species, ROS. And under normal circumstances, this molecule is really important in the cell-to-cell communication of these organelles. So a very important function under normal situation. When that cell becomes dysfunctional and becomes diseased or it's damaged, then the mitochondria become dysfunctional. They'll produce less ATP energy and they'll produce more of this reactive oxygen species molecule. And that becomes incredibly toxic and that causes the cell even more damage. So it's a very important process. Microtubules are also organelles within the cells, and they form like tram lines within the cell. And it's thought that the biophotons might be produced mainly by the mitochondria, and then they're along or pushed along these tram lines or microtubules to other regions of the cell. So they're mainly for transport. Now, all these folks have over the last 40 or 50 years have put together an argument that the production of biophotons is a distinct biological function. The main ones are that are as follows. It seems to be very, very closely linked to the production of reactive oxygen species, in other words, oxidative metabolism. And because oxidative metabolism is a very precise process of steps or molecular reactions, it's thought, well, they must subserve some sort of link to ROS. Secondly, they do cover a range of wavelengths from the shorter wavelengths of UV blue through to red and near infrared, longer wavelengths. Their intensity and their wavelength can change with health. And this is something Neuroscientists has been involved with over the years. For instance, if we look here, when they're healthy, they will tend to produce red to near infrared light. Or whereas once they become distressed or they're cancer cells, they'll tend to produce UV or blue light, the shorter wavelengths. The light is considered coherent, highly structured, and this is mainly put forward by Fritz Albert Popp here. So if it's coherent structures, you'd think it would be involved in something rather specific. And finally, the thing that really is interesting to me is that the mitochondria, these little organelles there, have light receptors. They have receptors for those wavelengths of light. Now, if you take each one of those 5 things individually, you'd think, there's not much of a story here. But when you take them collectively, all together, then there may be something there that the biophotons do subserve a particular function. Now, I'd just like to consider a little bit more this issue of the light receptors within the mitochondria and talk to you a little bit how the work I've done in the last 20 years or so using externally applied red to near infrared light treatment to treat various disease models. They work because I'm starting to think that the biophoton story is linked heavily with this red and near infrared light treatment. So I just want to tell you a little bit about this before we move on. So over the last year, many people have shown that if you apply these waves to the body, red to near, it has a big influence on cell function and survival. That photons can actually stimulate a chemical change in a cell, and that light energy can be converted to metabolic energy with a subsequent influence of cell function and survival. So what are the mechanisms? How does this work? Well, my friends, I'm glad you asked. What's happening here, if we light onto the brain here and we take a close-up snapshot of what's going on, the mitochondria, like I said before, have receptors for those wavelengths of light. And it's mainly found in a little molecule called cytochrome oxidase C, which is very important in the function of the mitochondria, together with the water within the mitochondria, and that's called interfacial water. And we know that water does absorb at least the infrared region of the light. And what that results is an increase in ATP energy. It drives the mitochondria to produce more energy. And if that cell is suffering stress, then it will reduce reactive oxygen species within that cell, so reduce the toxicity. Now, together with these short-term gains, energy gains, it does induce the expression of a number of genes, protective genes, stimulatory genes, good genes. In particular, stimulates the production of this molecule here, brain-derived neurotrophic factor, which has been argued that it's the single most important molecule in the brain because that substance, BDNF, promotes brain cell homeostasis, improves function and survival of brain neurons. So long-term changes as well. It also does other things. It stimulates the production of nitric oxide, which improves blood flow, and also VEGF expression, which is a molecule that induces angiogenesis, the production of new blood vessels. So all good things. It all produces a healthier cell all round. And what seems to be happening, this red and near infrared light seems to enhance mechanisms that the cell has inbuilt already. It just enhances the mechanisms within the cell. And it's not a targeted treatment. It can help any cell, potentially any cell that's in distress, not just brain. And some of them include, and others, on various disease models, on aging and neurodegenerative disease like Alzheimer's and Parkinson's disease, have shown that if you apply this external red to near infrared light on the brain, you'll get improvements. You'll get behavioral improvements and cell function survival improvements. If we look at aging models in rodents, you'll get improvements in cell survival and reductions in inflammation, together with cell function across the board, mitochondria, and cognitive improvement. In Alzheimer's disease, using various transgenic models, again, you'll get reduction in inflammation and improvements in the pathology, together with memory and cognitive improvements. Most of our work over that period has been on Parkinson's disease using a range of animal models, and we get similar things, improvement in movement of the animals, together with cell survival and reduction in inflammation. And when we use the gold standard of animal models, the non-human primate model, we got the same thing, reduction in clinical scores, improvement in movement, and cell survival and reduction in inflammation. So across the board in animal models, we have improvement using external light to the brain. Now, one issue while I was involved in this work was something I never really understood, and I never really talked about it in a talk when I was talking about the red to near infrared light treatment. I could never ever understand why the hell a cell so deep in the brain that would normally work in darkness would have light receptors. It just makes sense to me. And I tried to avoid it if it ever came up in a talk. Yeah, I never really had a ready reason for it until I heard about the biophoton story. And once I heard that biophoton story, I thought, the reason they have receptors is because they use light themselves to communicate with each other. And all we're doing when we're applying the external light is we're tapping into that biophoton system. So rather than this living in darkness and working in darkness, rather than that, we've got this sort of situation. The brain is full of lights and they're using that to communicate with each other. So just very briefly, how am I doing for time, Paul? Good?
Yeah.
Got a few minutes?
Okay.
All right. Okay. I'll just quickly go through what some very early when I joined Fond Clinitech, and you did pronounce that correctly, Paul. Fond Clinitech. I thought, well, let's start a program with looking at the photon story a little bit more in more detail. And some of the first questions was, do they define cell type? two main types of brain cells. We have our neurons, which are the main type of cells, and we have glial cells. And the first question was, does the biophoton emissions define cell type? Is it an indicator of cell health? Can we tell a cell if it's healthy or if it's stressed? And the thing that I was most interested and still am is, does an external application of red to near infrared light influence that biophoton emissions? And if it does, then that would go a long way to convincing me, at least, that is a main mechanism of this red and near infrared light application. With the details, I'm an anatomist. I don't know any of this clinical stuff, but just to point out that this really is the work of the wonderful postdoc working on this, and that's Jamie Ho-Kam. So I will talk this all her work. But basically, she got worked in a dark chamber, put the cells in a dark chamber, used a photomultiplier to see the biophotons. So the biophotons will come in, and that induces a release of electrons, and that's multiplied along the way. And then we've got a readout at the end, so we can see, and so we can measure the amount of biophotons there. We're mainly looking at intensity. We hope to look at wavelengths a little bit later. We looked at neurons, neuron-like cells, Neuro2A and astrocytes. We stressed them with a very powerful stressor, sodium triclosine, or the French call that eau de javel, which is very, very powerful stressor. And we used the external light, 670 to 810 red to near infrared at 2 doses, a lower dose and a higher dose, to see if there was any difference. Define cell type. And here is some readouts of neurons and astrocytes in green, neurons in green, astrocytes in purple. And you can see when they're healthy here and when they're stressed here, there's hardly any difference. We couldn't see any difference. So intensity in this situation really cannot distinguish between the two types of cells. And with this cartoon here, you can see the neuron here with this pattern of emission is pretty much the same as the astrocytes with the same colored emission, and whether they're healthy or they're stressed. Is it an indicator of cell health? So here we have our neuron graphs and our astrocyte graphs. This line here is the baseline activity, or the activity in the medium only. And this is healthy and stressed for both types of cells. And you can see with the healthy, they're just above the baseline. So yes, they are emitting some biophotons. But look at the change with them being stressed. Big increase for the neurons, big increase for the astrocytes. So in terms of health, you can tell in terms of intensity, there does seem to be a difference in their emission patterns. And you can see this with the changing color of, in this cartoon here, of the biophotons, that for both neurons and astrocytes, you do have this change. Now, what I mostly does external red and near infrared change or influence that? And when we looked at them at healthy and stress, and you look at the healthy situation at the low dose and the high dose here in the darker red, you could see here that for both the neurons and the astrocytes, no change. So on a healthy neuron and an astrocyte, the biophoton emissions, the intensity does not change with external red and near infrared light. But when they're stressed, we have a different situation. Here we have for neurons, this is when they're stressed. You could see that their levels go above the baseline, like I showed you earlier. And this is when we apply the low dose, and this is when we apply the high dose. And when we put two types of lights on there, you could see that their emissions increases quite substantially. So with both, so for the neurons, for the it goes the other way. There's a reduction here after red to near infrared light. So we do have a change, but we have a differential change dependent on the cell type. And this is shown in this cartoon here. You could see with the stress, it's blue, and it gets even darker blue, more stress with the red light, or I should say increase in emissions. But with the astrocytes, you've got the blue here and it's going to yellow. You have a decrease in intensity of emissions. And we could talk about what this may mean a little bit later, but I just want to highlight that we do have a difference with red to near infrared light. So a quick summary in terms of cell type, really can't tell the difference between the two types of cells based on intensity of emissions. We may get a difference with wavelengths, but in terms of intensity of emissions, can't tell the difference between the cell type. Under stress, we can clearly tell the difference between a neuron and an astrocyte. So it could be a measure of cell health using the intensity. And when we apply the external light, we do have a change for neurons and astrocytes in terms of their intensity of emission, but they're different, with neurons increasing intensity and astrocytes decreasing. And what we're doing at the moment, we're trying to incorporate wavelength into our story. Stay tuned for that. Where's this all leading? I think most of you are familiar with these three gentlemen here. They were a big child growing up in the 60s. This is Captain Kirk, and this is Mr. Spock. He's 2IC. And this is the medical practitioner, Leonard McCoy, or Bones McCoy. And there was one episode where he had a patient, and there was clearly something with the brain and under the cranium, and he put this device on the individual's head, and there were lots of colored lights going all over the place, and McCoy could tell what was wrong with the patient, as it had a subarachnoid hemorrhoid, but he could tell with this device, just scanning it across like that. And I thought, when I heard about the biophoton story, I thought, wouldn't it be great if we had a device to stick on someone's head, like a series of photo multipliers, and that can tell us, that can detect these signature light patterns and tell us what's wrong with the brain. For instance, if it's healthy, it's more likely to do red and near infrared, and if it's dysfunctional, it's more likely to be UV or blue. So we could have an early system of detection of disease based on light emissions. And indeed, from Naroshi's group, it's just, I was very, very pleased that the wheels are in motion here, that she's developing such a device, and we're well on the way for something. The reality of Star Trek and Captain Kirk is in place, and I'm looking forward to discussing that a little bit more later. Just a quick thank to the people at FOND, Jamie Hokam, who's he's leading the biophoton project, the wonderful engineer that set up the and Malvina Bies is the lab manager who runs everything. Without her, the whole building would fall apart. This is Clinitec here. You can just see the Alps here, and Grenoble's on one side of the Alps. Turin, where I am now, is on the other side of the Alps. And the home of Juventus, we should mention that too, Paul, not just Fiat. It's the home of Juventus. And a special mention for this lady, Anne Liebert. I was having a cup of coffee with her in the mountains in Sydney, and she was talking about 300 things all at once. And she mentioned light produced from cells. And I said, hang on a minute, what did you just say? She goes, light produced from cells, biophotons. I said, what, Anne, what on earth are you talking about? I thought she was completely mad. But then when I went and read on about it, I thought, oh, no, wait a minute, there could be something here. There's something interesting here. So I owe my discovery of the biophoton world to Anne Liebert. So yes, so thank you so much for the invitation. I hope I haven't gone over time, and look forward to the discussion.
Well, thank you very much, John. That was very thought-provoking, and I've got a whole lot of questions, but I'm moderating this, so I'm going to sort of restrain myself a little bit. I would like to introduce Nirosha Murugan. She's been patiently listening to what you've been saying. She too, as you've mentioned, is working in this area. She has a Tier 2 Canada Research Chair in Biophysics from Algoma University, and she comes to us today from Waterloo, Ontario. We're almost spanning the globe here. And I think and hope that, John, you have some colleagues in Sydney who are joining us. And that's what we like to do. We like to make this a worldwide discussion. Niresh, I'm just going to bring you into the conversation. Perhaps you could ask John one or two questions, and if your own work overlaps what he has been doing, then maybe you can tell us a bit about that too.
Absolutely. And first of all, thank you, John, for this exciting conversation. I'm beaming here with excitement because and seeing other people get engaged with this and in their own way is very illuminating, and all the puns intended. So thank you for that. I do have quite a few questions, especially since I really like the way that you have integrated photobiomodulation with internal light. And one aspect, as someone who's been trying to bring this into mainstream science, especially in biological science, biomedical science, is the concept of intensity and patterning. When light is usually applied in photobiomodulation, is there, we think about neurons when they fire the action potential. There's information in the way that these electrical signals are firing. Should we be thinking about how light is applied into the brain in a patterned way to engage with these neural cells?
Dosage is the bane of any treatment. You find the right dose. How's the saying go? The dose makes the medicine. And part of the problem with red to near infrared light, by the way, I hate the term photobiomodulation. It was introduced, I think, by Hampton and I just hated it. But I don't mind if other people use it, but I tend to use red to near infrared light anyway. So yeah, dosage is such a problem. It's so dependent on how many mitochondria a particular cell has. So it depends on the system that you're applying it to. And what we do know is this so-called biphasic curve within it. At low doses, it won't work very well, and at very high doses, it won't work very well. And there seems to be this sweet spot in the middle. But the problem is trying to find that sweet spot in there. I guess it's just like applying a drug. You've got your receptors, stimulate your receptors. We have receptors in the mitochondria. But I think we're coming to terms with how these receptors respond to the light. And we're not really, we're sort of getting there, but it's very, very slow. And there's so much heterogeneity across the literature on what's the right dose for particular systems. It is very difficult. Part of the group at Grenoble are looking into this issue, and it's just a minefield. Trying to get. It's also complicated to some extent when you talk about the living organism. That yes, you'll have a direct response with the light going directly on the cells, so you'll have that. But you've also got a systemic response. So we also know that it's doing something to the circulation. It's activating molecules in the circulation, whether it be immunes, free-floating mitochondria in the blood. So it's having effect on them. So how that's contributing to the whole story, well, we don't really know. So it's complicated wherever you can. I was just lucky 15 years ago when I first put the light on a Parkinson mouse. My student said to me, "Well, how much of this thing do I use?" I said, "How long does the machine go on for?" She goes, 90 seconds. I said, "Oh, do it for 90 seconds, see what we get. And it just so happened that was the sweet spot.
Yeah.
So dosage is a big problem.
Could I
Yeah,
just
and
interrupt the
oh, sorry,
exchange
go ahead.
just for a bit of clarification? Because one of the things that's puzzled me, and we've got some questions coming in, so perhaps you can clarify this first. Um, uh, so how is it that these photons can penetrate the skull? John, you showed this in the next slide, patients with some sort of helmet or something attached to the outsides of their heads. Do these photons get out? How do you know they're not just coming from the hair follicles or something? Yeah, how do you know they're coming from inside?
All I can say to you is we've done simulations showing that yes, both near infrared travels a little bit deeper, but the red and both near infrared can reach certainly the superficial regions of the cortex of the brain. So just the superficial, maybe 10 millimeters within the brain. That's one thing, but we have hard evidence. We have clear EEG, brainwave change, alpha, beta, gammas. They all change with response in response to transcranial light application, and fMRI. It's very clear. It will change the pattern of brain activity, pattern of brain connectivity across. It will also change blood flow and ATP levels in the brain.
If we stand under an infrared lamp, it could lead to a mood change.
Yeah, I know, and the psychiatrists are getting very, very interested in this now, because they're applying it to frontal areas of cortex here, and people that are clinically depressed show improvements.
So I do want to go down the clinical path with both of you in a moment, but I'm itching to just sort of get in because lots of people are interested in the following thing. So I'm a physicist, and light, means photons, and then what we would like to know is, are these just any old photons, or you mentioned the word coherent, John, earlier on, but establishing coherence and quantum properties like entanglement are not easy to demonstrate. But it would be enormously significant, because there are some people who think that quantum effects in the brain are linked to consciousness in some way, and we've got questions about consciousness, all very speculative areas. I just want to say, and we'll come back to this, that if you think quantum effects in the brain are mediated by electrons or ions or something of that sort, then quantum decoherence is known to be extremely rapid. So there's a lot of skepticism that that is the case. But photons would seem to evade that problem. Is it conceivable that there's a communication network, a secondary network going on in the brain of information exchange with quantum coherence or without it? If you've got photons flitting around between cells in the brain, is that part of understanding brain activity, understanding ultimately consciousness? I'm throwing out a lot of ideas at once here. It seems to me that the most solid ground that you're on is the therapeutic aspects, because whatever it is that's going on in the brain, we can argue about, and that's interesting for physicists. But if it works for therapy or for diagnosis, then that really opens up a whole new area of clinical practice. So could I ask you both to say, how close are we to doing actually using light in the clinic in this way? I know some has already been done. Rosha, could you tell us a bit about your work in that area?
Absolutely. I think so one of the most convincing data that we found was with the human population, where we looked at light emission exactly in the center type device that we have across the head, in conjunction with EEGs. And going into the study, I was very convinced that, when you have more activity based on cognitive states, you should have more photon emission. But in fact, we actually find quite the opposite. So that kind of gives us this idea that perhaps, yes, there's more activity, more photons being produced, But if it's involved in optical communication, then it's going to be propagated within the system and may not come out of the skull. So to answer your question, the first question you had there, Paul, about photons coming out of the skull, I mean, it's possible. And we have a diverse population of people with different hair color, different ages, and absorption rates. And we've also done some
some with no hair, some with no hair.
Yes.
Some with no, those are our best subjects, actually, I should say. And we are also doing some skull studies from animals to show, like, you know, that different regions of the brain of the skull does have different intensities of photons that can come out. So that's one things that we have validated. And then to really get into this quantum coherence idea, which is quite exciting. John, you had alluded to the microtubules, and I think there's should be some discussion about there. Microtubules have often been implicated in consciousness and emergence of conscious properties in terms of their metamaterials properties. And I think these photons, and this is something that we're actively doing right now, is looking at optical communication. If you put in a certain amount of light into axons from spinal cords, how much light do you get out? Are they scattered? Are they absorbed? What are the properties of these? So I think these kinds of studies will help us validate some of the near infrared studies. If light goes in, what happens to it? Like, is it getting absorbed? Is it just local? Is it being moved? And these are some questions that I think will help answer the quantum coherence questions.
Could I just interrupt again, because one of our participants, Steve Ruff, wants to challenge the idea that photons penetrate the skull. He says bone is opaque. These wavelengths, light will penetrate only nanometers, not millimeters. So I don't know enough about light shining through bones. I know when I was a child, I used to go under the blankets with a torch and flashlight, that would be called here, and switch it on, and it would shine through my hand from the palm to the other side. So I know that soft tissue certainly transmits light, but bone it does seem rather extraordinary.
Well, the bone hasn't been seen as a problem in the light community, and like I said, we've got the evidence that it does go through because it changes brain activity, and people have measured.
Is it to just get a chunk of bone and put an infrared source on?
Well, I've done it myself. I've done it myself.
We have done it too. I mean, and I can show you some data from pig skulls all the way down to cows to mice. Light can penetrate and I think what we are thinking about is that affecting the cortical layer. It's not going to go all the way to deep structures, but it's much like a network. You affect, like a domino, the cortical neurals networks, and then that propagates inwards and outwards. So there's clear evidence that light can propagate the brain, the skull.
I'm just going to throw out one other suggestion, and there's so many questions that I should really get to those. That has anybody looked at the simple system like the organism C. elegans, where its entire neural system has been mapped in detail to see whether this idea of photons being part of cognitive or brain communication was wired in right from the start, from the most primitive organisms. There you might be the chance of actually mapping the, I don't know what we call it, we've got the connectome, and what we do, the photone doesn't sound quite right. But if we imagine there is a photon network, could you make a start with something like C. elegans?
People have done that sort of work and found an effect, at least with the external application. Neuroscientists, I think there's also been explorations of the biophoton world at using simpler systems. Yes?
Yeah, I mean, I'm not sure if people have mapped it into the specificity that you're saying, Paul. I think cells and neural hippocampal slices, so brain slices, to kind of get through that penetration issue but still maintain network has been done, but it's something to try. The issue that I would have with C. elegans is they're in water, so absorption and diffraction of light into water-like systems might be I'm not sure how we could extrapolate that into rodents, but something to think about for sure.
So we had some questions emailed in ahead of time, one from Barbara Temple about biophoton entanglement. Now, obviously, that would be enormously exciting if that were to be the case. Any conjectures about how that might influence brain function?
Tangle. Sorry, what was that last word?
Entanglement, where
Entanglement.
so this is a concept that goes back to Einstein in 1935, where so the photons are linked by what he called spooky action at a distance, and this is the basis of quantum teleportation and the quantum internet and all these exciting things that are coming soon that the engineers are going to give us. But if something like that's going on in the brain, then that would be enormously significant. Do either of you have anything to say about entanglement, or shall we move on?
I mean, I think it's something I don't know how to even begin testing that, but it might explain some of the efficiency gaps that people are having a hard time explaining with the brain and how much information can be processed. So optical communication would be something to explore within the brain, which is something I'm really passionate about. So if anyone has any ideas on how to do this, I'm all ears.
Okay, that's a job for the future. Now, Karen Boone is asking about failing memory and dementia. And is there a signature there, an optical signature? Does the light, is there a change in the way that the light is being used in the brain, or the amount of light energy? Can that correlate with failing memory?
I mean, I can comment a little bit on that. John, do you want to go first a little bit?
No, you go.
So from our own work, we are now moving our brain helmet for what we call photoencephalography, where we're pairing the optical and the electrical communication into the wavelength I mentioned. But there's also other studies that have been done in terms of from Jaipai Dai, who has shown shifting in wavelengths based on aging. So the brain itself can shift towards the red wavelengths as you age. So there is some level of wavelength shifts that can be explored, and with our own stuff seeing in cancer stress states and what you're seeing, John, as well, there could be signatures of brain state. That's what we're hoping to see with our helmets.
So you get more of a shift to the red and near infrared with age.
With age,
really?
That's correct. Yes.
One would have thought it would have been more to blue and ultraviolet. Why do you think that?
I'm not sure. I mean, I don't know. I don't know if it has to do with penetration, if more of the UV and the infrared is being used. There is, again, going into the idea of consciousness, that the microtubules absorb in the UV, they absorb UV photons. So it could be that there's like this interplay between what's being used and what's actually being emitted, what we're actually measuring. And those are two different questions that need to be explored.
Or it could be with stress. It could be with the stress that the red and near infrared light is more common in that brain, because it's helping. They're helping themselves to repair each other, if you know what I mean.
Inflammation, perhaps, like, as we age, that's something that's a good point with mitobiogenesis. As you age, those rates also change. So there could be some sort of correlation there as well.
Okay.
Okay, we've got to move on because there's so many questions. Interesting second question from Barbara Temple here, because she talks about seasonal affective disorder, SAD. And that's something that I suffered from living in the northeast of England, where it's often foggy and dark. And I moved from there to Australia, which is certainly better from the point of view of sunlight. And now I live mainly in Arizona, where you're lucky if you ever have a day that doesn't have any sunshine. So I no longer suffer from SAD, but a lot of people do. And if light really does affect people's moods, I've always tended to think that's more of a psychological thing, but could it be a direct physical thing?
Mm. Well, they say that the the sunlight has, of course, has the broad constellation of wavelengths, and the blue component in there, particularly in the mornings, does improve your mood. It's not so good at night, of course, when you're looking at computer screens and telephones, and it sort of stimulates your mood and your attention and so forth. But during that morning shift, that the amount of blue light in the sunlight does have an effect of our mood. And even the transcranial work with the red and near infrared light, my colleagues in Australia, Catherine Hamilton and Ron Brown, have shown that when in Parkinson patient at least, that their mood improves, and their level of anxiety reduces, and their get up and go, as Catherine would say, is improved. So it has a big impact on general states of mood.
Okay, now here's something I didn't know. Called our anonymous attendee says that astronauts find that cuts heal better in space. I know that their feet become very smooth because they're not walking much. Cuts heal better. And chiropractors are offering infrared treatment for inflammation based on that finding. So could that be connected in any way with what you've been talking about?
Well, physios have been using red to near infrared light for decades, and in fact, Andre Mester, who was considered the father of red to near infrared light treatment, would have in his clinic he had a ruby laser, and he would use it to treat inflammation and wounds, and indeed the first applications were on this issue, on wound healing. Dentists use it. They have it in their clinics, and they would apply it to teeth that have been extracted to help the healing process.
It's also pretty common and popular right now in the cosmetic world, like face masks that have these red lasers, red lights on them. So it's pretty pervasive now, this use of red lights, and that's also in the racehorse, like equine. A lot of these really expensive race horses after a race have these red light therapies to reduce their inflammation.
So now
people have been using it to grow hair too, but it hasn't worked for me.
So your conversation prompted me to wonder what Tawfiq Naji is also wondering, that if infrared photons can have such a dramatic effect on the brain, should we be a bit more concerned about things like X-rays and MRI and other techniques? You think, well, they would have an even bigger effect on the brain. We tend to sort of we're told, don't worry about it. It won't affect your brain. But it's pause for thought, isn't it? If infrared photons can have such a big effect.
But they're good effects.
I mean, with MRI,
they're good effects.
Right,
you know?
but X-rays going through your head doesn't sound like a very good idea. But we're reassured that it will do no harm. It might give you cancer, but it's not going to affect your cognitive abilities. You don't have a CT scan and find that you're confused for several days. Doesn't seem to have that effect, and yet infrared photons do. It seems a bit mysterious.
well, we've been doing some applied magnetic field work to kind of show the transcranial magnetic field stimulation effects that people know that can improve moods, OCD, and psychiatric symptoms there. And we see that with a patterned magnetic field in low frequency ranges, less than 100 hertz, you get increases in photon emission. So they respond to they being the neurons, respond to applied electromagnetic fields. So based on like at least the MRI, it should respond, but these intensities are very large and also for a short amount of time. So long-lasting effects, I'm not sure, but definitely it will be doing something.
So Ryan Thomas is asking about epileptic seizures. And it's known that these can be induced by flashing lights. But is there a connection there with your work? Are either of you working on epilepsy?
I've sort of dabbled in it a little bit, and we do know that at least across the wavelengths that I've been talking about, red to near infrared light, it does suppress seizure activity, and we've seen that in a range of animal models, and it's just starting to be applied to humans. So yeah, I think the wavelengths have a big part of the effect it might have. So yeah, the other thing I wanted to talk about is the frequency. 40 hertz seems to be this magical number, and most transcranial devices now are built around a 40 hertz frequency. So you do have that flashing there, and it seems to be very beneficial for brain function. Narosha, I don't know if you'd like to comment on that a little bit more.
I would love to comment on that. Earl Miller has published this fantastic work showing like gamma oscillations in that frequency band is fundamental to brain oscillations in terms of the default mode network, what identifies us as individuals. And we see that also with any kind of applied stimulus light, magnetic fields. So there's this baseline frequency that our brain seems to respond to. And that you mentioned frequency, and I wanted to also kind of maybe let you know, like in your data that you had shown in your neurons versus astrocytes, the intensity is very exciting, but I would love to see if there's a frequency shift in terms of communication, because that opposing effects that you find with higher and lower, it could be that the neurons in the astrocytes are talking to each other in a sort of like polarized way in terms of the frequency band. So I think that'd be really exciting to look at. Happy
good
to
thought.
connect with
Yeah, good thought. Yeah.
We're running out of time, but I would just like to throw in one last thought. Back in the middle of last year, and I'm not going to say summer because it was winter in Australia, and that's where John and I met for a cup of coffee and began our conversations on this. And on that same occasion, I did an event in Sydney Opera House on the nature of consciousness with somebody from Victoria, from Melbourne, working on what he called dish brains. Getting brain cells in a dish, like a Petri dish, and sort of getting them to wire themselves up and seeing what they could do in terms of information processing and so on. So our conversation so far has been about, living animals, people in particular, and what's going on inside their heads, and how can you tell from transcranial work and so on. But I would be itching to just sort of chop up a bit of brain tissue or put it in a dish and see if there are photons being emitted or transferred. In other words, can you begin to map a photon communication network just by looking at sections of brains in dishes? So that's my last thought. We have about 1 minute. So would either of you like
all
to?
All yours, Narosha.
Yeah, I mean, I'm really excited. So I wish we had more time because this is exactly what we're doing. We have cell cultures of neurons, cortical neurons on microelectrode arrays, on nice lawns of neurons, and stimulating them with pulsed electricity and seeing what the photon emissions are. So so much cool stuff is happening in this space, and I just, with more time and people excited, I think one aspect you brought up, John, that I think might be good to end on is this bridging between the physics and the biology, and kind of there's a lot of in the biomedical community who understand biology but is missing this physical dimension, especially things like opacity and thickness and quantum entanglement. And so I feel like we need to work together somehow to bring the physics in to map, theoretically quantify, model all these work. And so that's what I'd like to say. We're doing it. We need support. We need to work together. Let's do this.
Well,
Yes, and talking to you, Paul, at that cup of coffee in Broadway was, well, you know, it was like it really opened my eyes. Goodness me, I'm talking to a physicist.
Yes.
Well, there are a lot of us about, and many of us are interested in what's going on in the brain and in these deep foundational questions in quantum mechanics, consciousness, and biophysics generally. And so we've explored a whole new world where these subjects overlap. And I particularly want to thank you, both of you, John and Nirosha, for contributing to this fascinating discussion. And of course, thank our audience and all those who sent in their questions. I hope you found it informative and entertaining. And I do ask you all to keep an eye out for our Thinking Beyond series next semester. We'll be starting up again at the end of August. So it just remains for me to thank everybody else and say good night or good morning in your case, John. Goodbye.
Thank you. Good morning. Good morning. Thank you again.